I'll introduce you. Hello everyone, welcome to another week of Soul Seminars. Today we have Leonie Moyle from the Indiana University. She has done a lot in Solanaceae. Her lab has worked on Solanum and also Hatamala, but also she's done work in other different species, considering the model system of flies. She's been very prolific in her research. Just this past year, she's published in Molecular Ecology, Evolution Ecology, Journal of Hereditary, and Evolutionary Letters. Today, Leonie will be talking about the history and origin of the natural variation in the charismatic wild tomatillo, Solanum pimpinellifolium, and its close relatives in the Galápagos. And then when you're ready, you can share your screen.
All right, let me see. I'm going to share my screen. And I'm going to go to this view. All right. Thank you, Chelsea, so much for that introduction and for the reminder that sometimes species names are a bit of a challenge, aren't they? Okay, I'm so pleased to be here. I have not been on the live versions of any of these, with the exception of the last one, but I've enjoyed very much going back and watching the YouTube versions of them. So hopefully this will be of interest to people here today. I very much appreciate both organizers putting this together and inviting me to talk. Okay, so let's get down to it. I actually changed my title a little bit because the other one was just so long-winded, although I gather that this one looks a bit long-winded too, doesn't it? Okay, so I'm going to talk today about some recent history and evolution of this charismatic wild species, Solanum pimpinellifolium, on the Galápagos. And the sort of subtitle of this is an evolutionary drama. So we'll get to why that is in a minute. Okay, so as Chelsea pointed out, one of the things that I'm really interested in my lab and the work that we do is working on diversification. So we're really interested in the process of how new species are formed and how new traits are formed. And we look at two different aspects, as do many people interested in these processes. We're interested in the pattern of diversification. So what's the order in which species or lineages diverge and how fast do they do it, and also the process. So what are the forces that are responsible for that divergence, and what are the underlying mechanisms of that change in terms of traits and genetics? So as Chelsea mentioned, we've worked in lots of different systems, but the one that I'm really passionate about and have worked on for the longest with these kinds of questions is obviously the Solanaceae. So that's why we're here. The Solanaceae is obviously a really cool group that attracts lots of interest because, as you can see here, it has a lot of economically important species in it. So as a result, it attracts funding and attention from lots of different sectors, including people interested in economic aspects of botany. But most of the people here also understand that it's actually really interesting, like diverse from a reproductive, from an ecological, and from an evolutionary perspective. And I think lots of people involved in this seminar series, including the last 4 speakers we've had, really been focused on the amazing diversity that we see. So I am really interested in the diversity, and obviously that's one of the reasons that I'm working in this group. But one of the things that I have learned is that it's a bit of a troubling group sometimes too. It can be both very beautiful, but it's also a bit dramatic. It's kind of a dramatic group. So the things that I've learned, I've worked in this group for maybe 15, 16, 17 years, something like that. And it's taught me a few lessons about how dramatic this group is. So what do I mean? If I think about this family, it's sort of steeped in evolutionary drama, not in a sort of not in the sense that some groups are in terms of their flamboyance. So if you think of, let's say passion flowers, Passiflora, that's very flamboyant as a group. I don't think of Solanaceae as a group as sort of flamboyant, but it's definitely dramatic. It has really dynamic, rapidly evolving lineages that are essentially running around the entire planet and speciating like crazy. As part of that process, that history is often really, really complex. And sometimes aspects of that history are hidden and don't really emerge, sometimes to your chagrin, until you're actually deeply steeped in trying to understand what's going on. But despite that, it sort of has a very common theme, and that's that natural selection is really dominant driving force. If you think about diversification in this group, you think about the beautiful trait, molecular, chemical diversity, it's really clear that selection is important. So this is the kind of drama that I'm talking about. It's sort of thrown up lots of unusual expectations for us when we worked in this group, and it's really an interesting group to work on because of that drama. Okay, the other thing that I think is really interesting about it is I think that drama, those processes really are reiterated across lots of different stages. So I'm gonna make myself small up here. There are lots of different scales at which you see those processes, both macroevolutionarily across multiple millions of years, microevolutionarily within species, and even in contemporary evolution. And the great thing about this group is being able to examine these processes across these different scales. So one of the macro scales that we've worked out is sort of looking at across lots of different species at a much broader scale, thinking about, for example, using whole genome analyses to look at relationships among these different groups. That's some of the work that we've done, but mostly it's been focused down on individual groups. So one of the groups that Chelsea mentioned is Haltomata. This is a really, actually it is sort of a flamboyant group, but also a dramatic group that Meng Wu and Jamie Costume in my lab, both former graduate students, worked on developing in terms of thinking about these questions. And it's a system that Jamie will continue on in her own independent research. So I won't talk about this group today. What I will do is instead focus down on the group that I've done the most work in, and that's the wild tomato group, Solanum section lycopersicum. So as you can see from these images from Euros Peralta's paper, it's a really beautiful group, and it's also really diverse, and it has its own drama as well. It is a fairly small clade within Solanum. Solanum is obviously incredibly diverse. This one is a kind of a dinky clade, I suppose, within it. It has about 13 closely related species, and the reason that I'm interested in it, and many others are, is that it's really geographically variable. It has lots of climate and ecological variation in its natural range in South America. And that leads to lots of morphology and functional diversification. And the first thing I was interested in studying this group is the evolution of reproductive isolation. Those are also really interesting. If you're interested in genetics too, it's great because it's genome enabled because of the tomato and all sorts of other resources that have been developed since then. Okay, so this group is the group that really taught me about the drama here. So in the wild tomatoes, we see all of these lessons about dynamism and complex history and natural selection. So in some of the work that we've done, we've sort of described the fact that diversification in this group is really rapid. You've got these dozen species in about 2.5 million years. And as a result of that rapid diversification, you've got all sorts of complexity underlying it. So instead of having nice, simple splitting patterns, we have a lot of complexity, a lot of gene tree discordance, a lot of shared ancestry across lots of different species. And we also have complexity in terms of integration. So recently, we published a paper that showed that integration is pretty common amongst species, even pretty well-formed species. Even though it's a small amount of introgress loci, it happens quite frequently. Nonetheless, there are clear elements that suggest that natural selection is acting actively on multiple different elements of this genetic complexity. So even though we have a complex history, we still have a really strong, flexible reaction to natural selection to produce this trait variation. So at this macroevolutionary scale, you can see all of those different elements, but I'm not actually going to focus on macroevolution. Instead, today, I'm going to focus down and think more about a microevolutionary scale, really more on tens of thousands of years and actually on hundreds of years. So let me tell you about the group that I'm going to talk about, and that's the group that contains the colored fruited species. So most of you know that the ripe fruit color of wild tomatoes is often green, or it has some other cool colors as you can see in this picture here. But the color fruited species are really limited to this group up here that contains the domesticated tomato and its closest relatives. Among those close relatives, there's a sort of a a group that's really interesting that is limited to the Galapagos Islands. So these are the players in this sort of drama that we're going to talk about today. These island species are found on the Galapagos. They've been studied for dozens of years. You can see up here in the top panel, that's actually a genetic analysis from Charlie Rick back in the 70s. There's 2 species. They didn't used to be. There used to be maybe 1 species or maybe not even any species. But of course, our description of species changes over time, as Sandy well knows. And actually, it was Sarah Darren's work that eventually described 2 different species on the islands, 2 different clades here. They have somewhat different characters, including colour of their fruits. So the thing that's distinctive about these island species, you can see that here's what they look like if you grow them in a greenhouse, is that they have orange or yellow fruits, and that's quite uncommon. The other coloured fruited species are red, or as I said, green or some variant of green. So these are the sort of supporting players in this mini drama. The actual protagonist in the drama is this species, Solanum pimpinellifolium, and it's also another member of this red-fruited clade. You can see its red fruits here. Here's the variation that if we grow up different accessions from in the species, you can see some of the variation in ripe fruit size, although the colour is pretty uniform. It is almost certainly always red in a normal accession of pimpinellifolium. So this species is, as you can see, quite beautiful. It's similar to the small red-fruited domesticated tomato, and closely related to them. And it also is really diverse. It has interesting ecogeographical distribution, mostly in Ecuador and Peru. And it has literally hundreds of collections, many of which are lodged in germplasm banks like the TGRC on UC Davis's campus. And a lot of that effort has come from historical collections that were made by lots of different researchers, including people like Charlie Rick. So many of these accessions have been historically collected, partly because of their different diversity, their diversity across ecogeographical clines. And you can see on the right-hand side of the screen, there's sort of descriptions, a graphical description of some of the really interesting environmental variation we have in the species range. As a result of that, we also have lots of different trait diversity, and that's another reason that people have been interested in studying this group. In some senses, lots of our research now is essentially just derivative of work that people like Charlie Rick and others have been doing and had done for decades. So it's describing natural phenotypic variation. Here you can see leaflets. There's also fruit. We've studied other kinds of genetic variation, like elements of architecture, like plant height, or where and how many stomata you have on leaves. Okay, so it's it's really interestingly diverse in terms of its phenotypic variation. And it's also quite easy to, or interestingly diverse in terms of characterizing its genetic variation. So there have been many genetic studies in this system, all the way from Charlie Rick's analyses of allozyme variation, through to microsatellites, through to more and more complex and more sophisticated genetic analyses from multiple different groups. Here's an analysis from Matt Gibson, a graduate student, and me, that came out earlier this year in molecular ecology, where we took many loci, about I think 10 or 12,000 loci, and did a multi-locus analysis. And you can see that variation that we see can cluster into regions, and those regions are actually describing geographical regions. The genetic structure in the species follows spatial clines, and that's been described for decades. We certainly didn't discover that. In addition to that, though, you can combine it with other kinds of data and be a little bit more sophisticated and think about what elements of the environment are driving that natural variation, and we've described some of them. And even you can combine those data with SNP variation and get down to individual genetic variants and genes that might be associated with that. So we can be a little bit more sophisticated nowadays than back in the mid-century last century, when thinking about genetic variation in species. But it's a very cool species, and it has a lot of variation. But as you can see, this variation is limited to the mainland. It's meant to be a mainland species. That's how it's been initially described. Okay, so I don't want to talk more about that genetic variation, more about that variation in the mainland. Instead, I want to focus on a much more, so that all of this variation I just discussed is mostly over the last, let's say, less than 500,000 years in terms of dating, which is amazing and super responsive. But it's also a species like any other species that has ongoing evolution. And that's what I want to focus on today, some contemporary evolution in the species. And that contemporary evolution that's occurring on the Galapagos. Okay, so this species has its own dramas, and one of the dramas is that it's escaped from its range on mainland Ecuador and has apparently started to run rife on the Galapagos. So let me set the scene for this particular drama involving this species. And the scene is focused around some research that we have done in the last couple of years, thinking about actually thinking about the endemic species. And this work has been done, in fact, most of the work I'll talk about today has been done by my amazing graduate student, Matt Gibson. Here's Matt actually in the Galapagos, looking very fieldy. And this work has been done in collaboration with Maria de Lourdes Torres at USFQ in Quito in Ecuador, who's our amazing collaborator and has been enormously helpful with both ideas and just the practicalities of doing research in Ecuador, which has its own challenges. Okay, so after we started thinking about doing research together, one of the questions that came up was thinking about the endemic species on the Galapagos. And as part of thinking about what's happening in terms of dynamic processes on the Galapagos, Maria is very interested in hybridization and introductions on the Galapagos in other groups, including guava. We talked about perhaps going down and resurveying the endemic species that occur on the Galapagos and seeing what's been happening in terms of their population dynamics that hadn't really been looked at for maybe a decade and a half. So Matt's goal was to go down there in the summers of 2018 and 2019, go to the 3 largest islands of the Galapagos and revisit a set of historical collection sites both from last century, but also into this century, done by other researchers, including Sarah Darwin and others, and see whether we could refine some of these species. So this is what he did. He went back and looked at these sites. And this is what he found. In terms of endemics, there's cheesmaniae, there's galapagense, and he went back to historical sites and here's what he found. So he looked for 26 of those sites and he only managed to refine 4 of them. He also found 2 new populations. The 4 that you can see are highlighted with the carrots. All of the other ones are no longer present. And then there's 2 round populations, which are in the lower left panel. Those are 2 new sites. And he also refound a single hybrid site. So there's already something interesting going on in terms of the dynamics of the endemic species. Many of those populations we couldn't refind. That hybrid site is really interesting. It actually was previously described by Sarah Darwin in her incredibly extensive survey of the existence or the occurrence of these species on the Galapagos. So she had already described this population, which is why Matt went back there. And in fact, when he went back there, he could see individuals that looked like they were hybrids. Here's some pictures of them. Okay, so that's the story with the endemics. Despite going back and looking at a whole lot of sites that seem to, or that had been previously recorded to have these endemic species, many of them could not be refound. The story also includes Matt looking for non-endemic species, including Lycopersicum, the domesticated tomato, and pimpinellifolium. And the story for those species is really different. In actual fact, he both looked at historical sites, but also for new sites, and found tons. Okay, so here we have the endemics. These are the ones that are meant to be native there. And here we have all of these new non-endemic populations. He found 30 populations, 23 of them had never been described before, and some of these populations were big, really big, hundreds of individuals. And 20 of the 30 populations were pimpinellifolium, this wild species that's from the mainland and should not be occurring in large numbers and in many populations on the Galapagos. The second thing about pimpinellifolium that was really interesting was that it had polymorphic fruits. So not polymorphisms within individuals, but there were populations some of these sites, 7 out of the 20 sites, all of the which occurred on Santa Cruz of this pimpinellifolium, looked like pimpinellifolium, but had some individuals that had normal red fruits and some individuals that had orange fruits instead. And orange fruit color is not characteristic of pimpinellifolium. In fact, it's characteristic of the endemic species. Here you can see a picture of cheesmaniae, and it's beautiful orange fruits. Okay, so we had, in fact, a case of what looks like phenotypic convergence. Up to about 40% of some of these populations had individuals that instead of having red fruits had orange fruits, which is a really interesting observation and not one that had been really made in a broad survey previously on the Galápagos. Okay, so that's what we saw. Here's the scene. We saw invasion, and especially invasion, many more larger populations of pimpinellifolium. We saw evidence for hybridization, both between endemics, the two endemic Galápagos species, but also some suggestion perhaps that there might be hybridization between endemics and the non-endemic invasives. And we found this evidence for a phenotypic evolution, this color convergence in fruit color. Okay, so that's the scene. It obviously raises some questions, and that's what I'm going to talk about, answering the questions that raises. So some of the questions it raises is, what's the history of these species being introduced, these populations being introduced onto the islands? How many introductions were there? Where did they come from, and when did they occur? And then the observations that Matt made on the islands lead to some fear of consequences of that. What are the consequences for interspecies interactions in terms of especially hybridization, and in terms of the phenotypic evolution that seems to be happening on the Galápagos? Okay, so how do you answer those questions? So very briefly, you take your field observations and collections, and you can combine them with lots of other kinds of data, but the data that we combine them with here that I'll talk about today is a population genomic analysis, and then using a bunch of models, either pretty simple ones or model-based simple models or more complex computational models, you can make inferences about the origin and timing and the demography of that introduction and the evidence for hybridization and other kinds of trait variation. So that's what we did. Again, this is mostly Matt with Maria's help. I am a bystander, but appreciative one. We had a bunch of field observations and collections that Matt had made. So in terms of the island samples, we had 174 individuals from 13 different populations from 4 different species. It's the 2 island endemics, the pimpinellifolium, and some domesticated lineages or Lycopersicum lineages. And we took those with Maria's help. We managed to get permits to get DNA out of Ecuador, and we subjected them to reduced representation sequencing, dd-rad sequencing. Okay, so that's just a method that takes some subset of the genetic variation across the whole genome, sequences it up, and generates markers. And the reason we use that is that we were then able to pair those data with the data that we'd already collected for all of those mainland samples from pimpinellifolium, the data that I talked about before that's been published, and that Matt generated from mainland accessions. So we have a bunch of island samples, we have a bunch of accessions from the mainland from pimpinellifolium. And you can, I'll show you just some numbers, the details don't really matter, but you can generate quite a lot, even though it's not whole genome sequencing, which perhaps would be the gold standard for thinking about population genomics. You can generate a lot of loci that are quite helpful, and thousands to tens of thousands of single nucleotide polymorphisms that help you reconstruct and understand history across all of these different samples. Just to give you a sense of what that looks like, they're distributed across the chromosomes. These are actually data from the mainland samples. So you get a really good snapshot of history that isn't allozymes, and that can give you much more fine scale ideas about relatedness and about ancestry at different parts of the genome. Okay, so we had our field observations with the collections, we did a population genomic analysis, and we can look at variation both in the island invasive pimpinellifolium, the mainland pimpinellifolium, and the endemic species as well. And then subject all of those data to these simple and model-based inferences. Now, I'm not going to talk about all of these, but I'm just going to mention we threw a battery of models against some of these data, all for good reasons, not and I won't talk about all of them, but there's lots of different ways that you can turn these data around and think about using the fine scale genomic variation to think about these processes. Okay, so with those data, what can you do? Well, you can address these questions, and I'm gonna go through them, you know, one at a time mostly. So thinking about history, using those data, how do we address what the history of invasion was? How many there were, where they came from, and when? Well, the first thing you can do is essentially, oh, and I'm going to tell you the answer. Sorry, I forgot I was going to do that. So just so you know where I'm going, we have evidence for at least 3 introductions onto the Galápagos of pimpinellifolium, but really only one that led to this expansion of population. So what I'm calling one invasion. And it's almost certainly the origin of that invasion is almost certainly from Ecuador, and it's pretty recent. Okay, how do we infer those things? Well, as I said, using all of those genetic data, you can essentially generate these multivariate genotypes from thousands and thousands of SNPs across the genome and collapse all those data down using principal components and get an idea about where in principal component space your genotypes are falling. So that's one of the first things that we did with these data. So I'm going to show you what that looks like. So we have here our PC space. We have axes of variation that are explaining, the predominant variation in our data set across all of these SNPs, single nucleotide polymorphisms, across all of these genotypes with thousands of loci. And so I'll show you what that looks like. So you can see that we have a set of mainland samples. Those are the ones from the previous study. And they're broken down into 2 different geographical locations, Peru and Ecuador. Peru is in boxes, Ecuador is in diamonds. And you can see they sort of fall over on the right-hand side here of the plot. And then we have a set of island examples in circles, and it's pretty clear pretty fast that there's some structure to where those island samples are falling out. There are actually 3 different locations. 2 are singular. So we've got a pink and we've got sort of a maroon color one, and those are actually from single samples. So single original collections. One of them, this maroony colored one, was actually made in 1991. It's one of the accessions from the TGSC. That falls out by itself. One of them was one of Matt's accessions, single collection, single individual, that was collected in 2018, and that falls out too. And all of the rest of our samples, dozens and dozens of samples, all fall out together. Those are Matt's collections from 2018 and 2019. Okay, so that's the first hint that we might have 3 different things going on here. What else can you do to understand how these relationships are falling out? Well, you can visualize it as a tree. And so you wouldn't normally make a tree out of data from within a species, but it's a simple way of visualizing stuff. So what you can see here is essentially a Raxamel tree that Matt made with a subsample of the mainland data and then some sample of the island accessions or collections. And you can see down at the bottom here is all of those purple circles. They're basically all falling out together in a cluster, and those island pimpinellifolium genotypes are actually nested within a larger Ecuadorian clade. And then you can see the pink and the maroon one up there, those are separate again. They fall out somewhere else. They're not falling out with that cluster of purple. So most of those island pimpinellifolium genotypes are nested within really closely related to each other, and they're within an Ecuadorian clade, suggesting an Ecuadorian origin. Then pink over here, that other singular collection that Matt made, also looks like it has an Ecuadorian origin. And then the previously collected one from 1991 really looks like it comes from Peru originally. Okay, so based on that data, we think that this invasion came from Ecuador. Most of those samples, or those purple samples, come from Ecuador. There's also other data that support that. So one way of thinking about it would be to say, how distant are all of my Galápagos samples from either Ecuador or from Peru? And that's what these data say. So if you think about the average pairwise divergence across each of our different loci, obviously they're not very diverged when you compare all of the island populations, the invasive populations, to each other. They're all pretty closely related. And on average, they're less diverged from Ecuadorian populations than they are from Peruvian. Again, another piece of evidence that this is an Ecuadorian introduction. And perhaps it's a little bit easier to visualize here when you can sort of see the array of those samples across the range of pimpinellifolium, and you can see that's sort of a heat map. The darker the color, the closer, the less distant these mainland samples are from, on average, from the invasive individuals. And you can see that the closest relationships are up in Ecuador. So we also use a number of other modeling processes, including this very cute resampling model called Locator, which is recently published, and you can essentially use genetic data to say where did this thing come from. It's very good for new dispersal and new invasives, and that also says that almost all of our samples come from Ecuador, except for those two. One of those comes from Ecuador and the other one comes from Peru. So most of our data are pointing to this idea that we have 3 introductions. One is a major introduction that led to this invasion. Most of those individuals on the island are from that major introduction, and that came from Ecuador. How long ago did it happen? Well, you can say pretty confidently that it was pretty recent. Partly you can say that because there hasn't been a lot of records of pimpinellifolium on the Galápagos, pimpinellifolium proper on the Galápagos. Certainly not as many as we see right now, but there's also other pieces of data. So again, that pairwise divergence across all of these loci, I'm just going to draw your attention to that axis. That is not a lot of divergence. So you can, roughly speaking, translate that axis to a percent. So there's less than 0.2% divergence on average between all of the island invasive species, invasive individuals, and Ecuadorian populations. It's like really, really, really low sequence divergence, which suggests a really recent introduction. But there's other more sophisticated analysis. You can also think about pi, which is a measure of polymorphism in windows across the genome. And again, in the invasive pimpinellifolium populations, pi is really small, suggesting a really recent bottleneck. So there's lots of elements of the data that suggest both where it came from and how recently. You can also do more sophisticated analyses. So one of the analyses that you can run that Matt has done is use elements of the site frequency spectrum from some of those invasive populations in order to make inferences or estimate parameters in these populations. So this is using DADI, which is another way, which is one of these models. And when Matt reconstructs this with a single population from the islands, our most deeply sampled population, he infers that there was indeed a bottleneck, which is what you expect in an introduction, followed by an expansion. That's also shown by negative to GMSD in the island populations. And you can estimate timing as well of when this happened. So the timing is kind of interesting. Here's the best guess in terms of the optimal timing, and it's kind of long. So one of the interesting things about these models is that they can give you relatively unexpected answers, but they also have, especially if you don't have a lot of variation, and we don't have a lot of new diversity in the populations on the islands, they can have really high confidence intervals. So it suggests, the model here suggests that the introduction or the population bottleneck started somewhere around 2,000 generations ago and ended sometime around 52 generations ago with really big confidence intervals. Somewhere between 100 generations ago it started, and somewhere down to 36 generations ago it stopped. So these are broad. I actually think that they're probably overestimates but they're within the range of something that's to do with human introduction, which is what we think has happened here. Incidentally, if you run these models using the endemic samples instead, the two Galapagense and cheesmaniae, you see much older ages. You also see a bottleneck and an expansion, but that's happened much further in the past, which is what you would expect from an endemic set of species. Okay. So one final piece of data, and those of you who are eagle-eyed probably noticed this before, there's this really suspicious looking data point in the middle of Ecuador, up here in the Midlands of Ecuador. And that data point is from a collection, an accession that was collected in 1956 on the mainland. And you will actually see, if you look down in the PC plot, that that data point falls right in the middle of the island endemics. The island invasive pimpinellifolium. So the first time we saw that, I was like, oh, that's so great. That's the origin. That's where it came from. And I think now we're fairly convinced that that's probably not the case. It's so very closely related that it's probably a back colonization. It's an event that we believe is probably human mediated, where somebody brought back pimpinellifolium that had been introduced onto the islands back onto the mainland. And so that actually puts a bit of a boundary date-wise in terms of how they must have, that invasion must have occurred before 56 in order to do a back collection back on the mainland. So that's our influence right now. It would be really fun if that was the origin, but I don't believe so. Okay, nonetheless, those data together suggest a couple of different things. 3 different introductions at least. There are probably other samples that we haven't sequenced that may represent additional introductions, but only one of them has become invasive. And that introduction was probably from Ecuador, and it was probably really recent, sometime in the last couple of 100 years is the guess, and almost certainly associated with human colonization of the Galápagos. Okay, so what do those data tell us about this other downstream drama that might be happening on the Galápagos because of pimpinellifolium? What are the consequences of that introduction, and what can we learn using the data that we've already collected? Okay, so one of the consequences I talked about was this hint that there was hybridization and introgression going on, and we have good data to suggest now that that's absolutely true. There is evidence for hybridization and introgression that's as a consequence of this introduction. Okay, so what does that look like? So some of this hybridization isn't involving the introduced pimpinellifolium, it's actually involving the endemic species themselves. So back to that population that I mentioned previously that Matt had refound that originally was described by Sarah Darwin. That's on Isabella, and it's a population that has intermediate individuals that look like there may be hybrids between Galapagense and cheesmaniae, the 2 endemic species. So we can take our data and basically assess what's the status of these individuals in these populations. Are they in fact genetic hybrids? They appear to be morphological hybrids. And the answer is, yeah, they appear to be genetic hybrids too. So you can see, again, a multi-locus PCA up in the upper left-hand side that essentially shows individuals that seem to be hybrid or intermediate in the field in sort of a pinky salmon color. And they are in fact falling between the 2, cheesmaniae in blue and Galapagense in green. And if you do a reconstruction, or if you do an inference based on admixture proportions using fast structure, which is a very common way of thinking about relatedness among individuals and which proportions of the genome can be assigned to different ancestries, you can see here that in that population in MG120, there are individuals that appear to be just cheesmaniae, individuals that appear to be exclusively Galapagense, and then a bunch of hybrids again. And the hybrid indices that come out of that suggest pretty recent hybridization. Okay, so there's certainly dynamism in terms of even the island species themselves appear to be getting up to some shenanigans. Okay, but we also see hybridization between the introduced species and the endemics. Okay, so for example, again, this is a structure plot from fast structure showing admixture proportion for different individuals. So each bar, each column is a different individual, and the colors are its ancestry assignment, essentially. And you can see there's 3 different invasive pimpinellifolium populations, and primarily the ancestry of those populations is assigned to pimpinellifolium, brown in this case. But there is a site, MG115, that had mostly cheesmaniae, but some individuals that look like pimpinellifolium or something odd, something weird. So if you think and they should have looked odd, because in fact, we infer that a number of these individuals actually are hybrid. They have mixed ancestry in terms of both pimpinellifolium and cheesmaniae. So this is cheesmaniae is an endemic, pimpinellifolium is an invasive species. And here we have individuals that are showing a mosaic of those 2 different genomes. Actually, if you look at some of these individuals and you use another method called new hybrids, 4 of the 6 that seem to have some unusual admixture proportions are actually inferred to be either F2s or F1s. So this is really recent hybridization. It's the last 1 to 2 generations that's occurring in the field right now because of the introduction of this invasive species. Okay, so this is dynamic and dramatic, but we also see evidence not just that it's happening right now, but it's happened in the recent past, but not, last week, in the last couple of decades, presumably. Okay, so I'm going to draw your attention to MG114 because I'm going to talk about that population. In the fast structure analysis, that comes out as having, largely pimpinellifolium ancestry. But different analysis methods use different approaches in order to infer ancestry, and some of them are more or less sensitive to different kinds of assumptions. So what we wanted to do was think about some of these other populations and say, okay, well, we don't have screaming signals of integration. These aren't F1s or F2s, but do we have any other piece of evidence that perhaps there's been some hybridization as well? So we took populations like MG114 and used other kinds of approaches to address that. And one of these approaches was to use an HMM, which is an algorithm that can essentially march along the chromosomes, and using the SNP variation, the polymorphisms we have along chromosomes in different individuals, essentially assign ancestry. So this is what the data look like from that MG114 population, and with each of the rows being a different individual. And you can see the ancestry assignments are either pimpinellifolium or cheesmaniae. So hopefully it jumps out to you. So this is on 100 KB windows. RAD-seq data is not the greatest data for these things, but it's adequate if you have a large enough window, and it gives you a sense of what might be happening. Ideally, whole genome sequencing, if people want to give me money for that, I would love it. Next panel, you can sign me up. Okay, so I do want to point out what these data show though. Okay, so you can see that the HMM, the model, is predicting some mixed ancestry here. In actual fact, 13 out of these 15 individuals that really look pimpinellifolium-ish have some evidence of ancestry. And on average, it's about 4% of their genome looks like it has ancestry from the endemic species. So that's evidence of gene movement from the endemic into the invasive. But your eye is not letting you down. It's clear that that ancestry is concentrated in certain parts of the genome. You can see on chromosome 3, there's a pretty chunky haplotype sitting there, and that's a frequency of about 40% among our individuals. And on chromosome 6, we have another fairly chunky piece of the genome that seems to have cheesmaniae ancestry, and that's at about a frequency of 0.2 in the population. So it's not uniformly distributed, it seems to be concentrated in areas, and the distribution of these chunks here suggests that some of these individuals are not independent of each other. They're probably siblings, they may be siblings, they may be cousins, right? These are not independent introgression events. Nonetheless, focused on these 2 regions. Okay, so looking at a simple fast structure, we don't see ancestry, but looking at a slightly more detailed, different method, we can pick out regions of the genome that seem to be introgressed in the recent past. And actually, if you make an inference, it's probably about a dozen generations that it would take back crossing to get about this much genome into that much background. So, you know, dozens of generations, but probably not longer than that. Okay, we can also look at other populations. So here's another population, MG117, where we also applied this HMM. And in that population, you can also see that we have evidence of mixed ancestry. In fact, all of the individuals in this population had some elements of mixed ancestry, again, from cheesmaniae. Again, it's about 4.5% of the genome, but again, it's not randomly distributed, or it's not distributed evenly across the genome. In actual fact, you can see again, there's a big haplotype on chromosome 6 at a frequency of about 40% in this population. Okay, so that's kind of fun. We have sort of cryptic introgression here. We have both, oh, sorry, so not all populations look like that. So the two I've shown you have about 3.5 to 4.5% cheesmaniae ancestry. We looked at least another population, and we inferred that it had about 0.5% of cheesmaniae ancestry. I don't really know what to make of that. My guess, or my inference, is that that's either misspecification of the model, or it's ancestral variation. I don't think that's very good evidence of introgression. There's no obvious components of the genome that are introgressed. So my inference right now is that that's probably just consistent with the kind of misspecification you would get using an HMM. It may not be evidence of even further ancestry in the past. Okay, so that's fun. We have a bunch of data on invasives and endemics, and we can actually show that things that look like they're hybrids are indeed hybrids, and in fact, individuals that don't necessarily look like they're hybrids also seem to have a hybrid ancestry a little bit, and that the ancestry is from the endemic species into the invasive individuals for the most part. Okay, how does all of this play into this phenotypic evolution that we see? This sort of dramatic shift in phenotype of fruits that we observed, that Matt observed in pimpinellifolium. Okay, so just to remind you, we have this evidence of phenotypic convergence. So pimpinellifolium is almost uniformly a red fruited species. There are very few examples on the mainland of evidence for orange fruit color. Cheesmaniae is orange fruited, but there's a bunch of sites on Santa Cruz where we have pimpinellifolium looking individuals. Their foliage, the shape of the plants and everything looks pimpinellifolium, but they have orange fruits. So what can we understand about what's causing this orange fruitness with the data that we have? Okay, well, a useful way of thinking about it is to think, where could that convergent phenotype come from? And there's a couple of different origins that could explain the convergent phenotype. One is that the pimpinellifolium on the Galápagos could just be reinventing something that's already happened. So I mentioned that there are some cases on the mainland where people have recorded orange fruited pimpinellifolium. There's like maybe 2 or 3 accessions that are described that have orange fruits. So it's possible that there was variation in the introduction itself. So it took an allele from the mainland and actually reinvented it on the island. Okay, we don't think that's the case. One of the reasons for that is all of the examples that at least I'm aware of orange fruited accessions, and they're exceedingly uncommon, less than 0.5% in pimpinellifolium, are from Peru. And the introduction we have is from Ecuador. So that's one reason. We have other reasons, which I'll talk about in a second. But I don't think that the source here was a reinvention. It wasn't an introduction of a standing allele that was already in the mainland. What are the other alternatives? Well, you can create an allele. So you can essentially have a de novo mutation. Think about the different genes that give rise to fruit color and mutate one of them and give yourself some orange fruits. So we know something about the alleles or the genes that cause fruit color variation in tomato. So this figure on the left-hand side was actually a figure that Stacey showed a few weeks ago in her talk, which is beautiful, and it describes how complicated some of the interactions are for the generation of carotenoids, even in tomato, where it's one of the best understood systems, carotenoids in fruit. Okay, so I'm going to be a coward and I'm going to take away that figure, which is beautiful and probably very descriptive. And I'm actually going to step back and use this other figure, which is good enough and simpler, from Parran and Vandernapp back in the 2000s. And that shows you the elements, some of which we understand contribute to fruit color variation. So what one could do if one was evolving orange fruits would be basically to mutate one of these elements of the non-carotenoid biosynthesis pathway to create new phenotypic variation. And so there are actually a number of different mutants that are really well understood in tomato, and here's an array of fruits, again from the TGRC site from Carl Jones when he was there, and you can see mutants that change fruit color here. Okay, so island pimpinellifolium could have just mutated one of these genes. It could be a de novo mutation. So that's a possibility. It's something that I think we need to explore more rigorously by essentially going in and sequencing out a bunch of these genes and confirming what their allelic state is and whether they have any evident mutations, either using sequence variation or expression or whatever other assay. I will, however, note that one of these is really interesting gene, and that's Psych-B. It's also known as the Beta Locus. That gene itself was first described in Galapagos tomatoes, it was in cheesmaniae. So it raises the possibility that instead of mutating, why mutate when you could just steal? So the third source of phenotypic convergence is basically theft. Instead of pimpinellifolium making a new allele, it could just steal an allele that we know exists in cheesmaniae at this locus. And that allele is a dominant allele, and when you mutate that gene, it leads to orange fruit color. So what we wanted to do was use our data to explore this idea of theft, trait theft. And one way to ask that question is cheesmaniae ancestry associated with fruit phenotypic variation in pimpinellifolium? With a particular focus on different genes in the carotenoid biosynthesis pathway, including ones that are known to cause variation among wild species, especially cheesmaniae. So that's what Matt did. He basically used an analysis. He went in and looked at regions of the genome that seemed to have CHS, cheesmaniae ancestry, and said, is that co-associated with fruit phenotypic variation? Okay, so I told you already that we have a couple of different populations that seem to have cheesmaniae ancestry at parts of their genome. I didn't tell you, however, that the two populations that I was talking about here are actually both polymorphic for fruit color. 114 and 117 are two of those populations where Matt found variable individuals, some with orange fruits and some with red. So you could actually look at the association when you have both the genotype and the phenotype. So that was our goal. So I'll start with data from MG117. You can see that the biggest region of CHS ancestry, of endemic ancestry, is that big chunk on chromosome 6. In actual fact, when you go in and you look at that chunk on chromosome 6, you can use the HMM to assign ancestry along a window of chromosome 6. So here's what those data look like. There's 3 different ancestry states. There's homozygous for PIMP, homozygous for the cheesmaniae allele, and heterozygote. And you can see there's a bunch of individuals that have different components across that region of chromosome 6. And in that region of chromosome 6 is a gene that's involved in carotenoid biosynthesis, Psych-B, the Beta Locus, which is awesome. Okay, so the question is, we have individuals that are homozygous and heterozygous for different allelic states at Psych-B. What do their fruits look like? Here's what their fruits look like. And it may not leap out to you immediately, but there's a perfect association between the presence of at least one cheesmaniae allele at Psych-B and orange fruits. So this we're taking as prima facie evidence that actually the orange fruit color in this population is probably because the individuals with orange fruits have ancestry at the Beta Locus from introgression from cheesmaniae. Fun. Okay, so ideally, so we will be following up as this inference later with additional work. But okay, we've got a causal locus, and I already told you that the other population that we looked at also has a big chunk of chromosome 6. So surely that is also the beta locus, but of course it isn't. So they look very similar here. There's not enough grain to be able to tell, but when you actually go down and look at the fine scale data on MG114, the beta locus is not introgressed from cheesmaniae into the background of pimpinellifolium. It's missing. Those are pimpinellifolium individuals at that locus. So we know it's not the beta locus there, or it couldn't be. So what else is happening? Well, let's look at chromosome 3. So we did a similar kind of analysis at chromosome 3, and that haplotype that I showed you is sitting over a region that contains Psi1, PSI1, which is another element of the carotenoid biosynthesis pathway. And if you squint, you should be able to tell that there's also perfect association between the allelic state at Psi1 and the fruit color of individuals carrying those loci. So Psi1 actually acts recessively, so you need 2 copies of cheesmaniae in order to have an orange fruit, if that's the causal locus. And there are individuals here that all individuals that are orange fruited, in fact, do have 2 copies of cheesmaniae at this locus. Okay, so our inference is that, in fact, we do have some thievery here. We've got pimpinellifolium that has essentially stolen allelic variation, not the same one. In one population, it's been variation at the beta locus, and in another population, it's been variation at R, in order to converge on this phenotype. So as I mentioned, we will be confirming this with other kinds of data, including manipulative experiments, but this, I think, is pretty good evidence that in 2 different cases, we've got introgression that's led to phenotypic convergence of this invasive species on an endemic, and with 2 different genes contributing to that. Okay, so I started with this sort of confusion of like, well, it seems something dramatic's going on the islands, what's happening? And what we have shown is using genomic data in combination with phenotypic data and observations in the field, you can actually draw some inferences about this sort of quite dramatic recent history. This introduction, expansion, a set of hybridizations, and in fact, a change in the phenotype of invasive individuals towards the island phenotypes. Okay, so this is just a way of saying that pimpinellifolium itself is showing these evolutionary dramas. It's dynamic, it's rapidly evolving, it has some complex history that's sometimes hard to see without some sophisticated methodology, and natural selection is the dominant driving force. Okay, but I didn't say anything about natural selection, so I'm just going to use the last 2 minutes to say something about natural selection, because I didn't, did I? So here's the question. We have convergence of fruit color. We have a red fruited species that's becoming orange fruited, or some number of individuals in these populations are orange fruited. And the question is there a reason for that? Is there a selective reason why one might want to converge on orange fruit color? So the classical explanation for fruit colors on the Galápagos has been focused around bottlenecking and the loss of red color because of drift. My feeling right now is that possibly is not the best explanation. That's possibly really a selective reason why it might be good to be orange. We don't know what it is. There's a number of different mechanisms. The proposal I'm going to throw out there, I'm happy to hear if other people think about these processes, is that there is possibly an advantage to being an orange or a yellow fruit colored species on the Galápagos rather than being red. That advantage could be mediated by abiotic factors, or it could be mediated by the preferences of things like dispersers. So here we have a Galápagos mockingbird. When Matt was on the islands, he saw them visiting populations and taking fruits. So it's possible that, that the selective pressure imposed by something like the preference of a disperser may be something that is leading to the shift in phenotypic variation in this invasive species. Regardless, hopefully I've shown you that you can understand what's going on in this really amazing group at all different kinds of levels, and it shows lots of different but very similar kinds of themes, no matter what diversification scale you're interested in. And we're really hoping to work in the future to fuse these scales together to think about things that happen at macroevolutionary scales and how they flow down to microevolution, and how you can also feed back up to macroevolutionary change via those processes. Okay, I need to acknowledge the people who really did the work. Very briefly, I mentioned some of the macroevolutionary stuff, and I have an amazing set of collaborators that contributed to that, but really the people contributing to the work that I talked about today are obviously Matt Gibson. He is a huge driving force in all of the stuff that I talked about. Maria Dolores Torres, our amazing collaborator in Quito, is also absolutely instrumental. And Yaniv Brandvain, a former IU person, now at Minnesota, was also instrumental in getting that HMM to work so that we could really think about fine scale patterns of ancestry. And with that, I'm happy to take any questions if you have any. And I guess I will stop sharing my screen in order to allow that to happen.
Thank you so much. That was a great talk. We already have some questions in the chat. I don't know if people want to unmute themselves or I can read them. The first one is Franco E. I'll give you a second if you want to ask your question. It's okay. Okay, so he said, very interesting considering that islands have complex geographical history. So he said iguanas are said to be older than islands. Could it have been reintroduced reintroductions that did not come from the mainland, but from islands that no longer exist?
Oh, that's a really interesting question. So it's sort of like it hopped across islands that now no longer exist and sort of got to where it is. I don't know the answer to that question. I mean, I certainly know the answer to the question with respect to pimpinellifolium. I don't think that that is the case. With the actual endemics themselves, what we know is that all of those red-fruited species or all of those color-fruited species are really closely related to each other. It would have to be a bit more sophisticated analysis to think about the timing of that. In terms of our inferences, it's pretty old, but it's certainly not older than the islands themselves. So from the data that we have from population genomics, it doesn't seem that there's evidence that perhaps comes from other organisms that there were islands in the past that have now disappeared. Our dating on the endemic species seems to be about how old you would expect them given the age of the Galápagos themselves. But it's something to think about, whether we can use our data to think more about that.
Question from that, is there a way to determine like how many generations it's old, like old it is like, what's your, I don't know, your standard error with estimating how old things are?
Yeah, so you're at yes. I mean, yes and no. I mean, part of our inferences are based on, well, all of the inferences are based on the presence and distribution of genetic variants, right? So they're all indirect inferences from the distribution of that genetic variation. So interpreting that data requires that you essentially feed in an expectation about what the generation time was. And then it will tell you how many generations in the past. So the more variation that you see currently, the more generations in the past. That's a simpler way of thinking about it. So the bounds on that can be really big if you don't have a lot of variation, because then the stochastic element of like mutations feeds into your predictions. And I think that's sort of what's going on with our data reconstructions. We don't have a lot of variation, and so it really matters whether you've got a specific SNP or not, how many generations you're inferring in the past. The further you get in the past, the less it's influenced by individual variants, and the more it looks like the whole reconstruction. And so I think it's because it's so recent, it's really, you have these massive confidence intervals on that. Is that a little clearer?
Yeah. And I think the next question is Zach Lippman, if you.
Hey,
Zach.
Leonie, how are you? How you doing? I'll distill it down. I'm just wondering whether those introgressions for color genes, the segments, the size that they are, maybe they're bringing in something else that's adaptive, that's completely separate from the color genes. You know, these haplotypes, you know, that can bring in not only that, but many other things.
Yeah, it's a great question. And the answer is, could be. I mean, there's massive numbers of genes. They're, I don't know, megabases and megabases long right now. So that's the reason one would want to test a selective hypothesis. So one way of doing that is essentially just get rid of everything else, just change the color and see whether that's the thing and manipulate it on the island. So the funnest thing would be, let's see whether there's actually a preference for orange food color. among
Yeah.
seed dispersers on the island. Then you're not worried about whatever else is coming in.
Right.
You basically say, let's put out a bunch of fruits and say, do the dispersers like orange fruits more than they like red fruits? I think that's a very simple sort of classical behavioral experiment to do. So yeah, the answer is we can't be absolutely certain, but it is sort of intriguing.
Yeah. Related to that, I'm wondering, like the recent paper from sunflower and those large haplotype natural balancers, do you see any evidence of very large inversions within this small group? Can you detect that with RAD-seq? Probably not.
We cannot really detect it with RAD-seq, but historically there isn't a lot of evidence for inversions from other kinds of work. So there may well be inversions, but I think the wild tomatoes are sort of interesting because they don't have so much dynamism in terms of like structural variation. I mean, there are some well-known ones.
Yeah.
But if there are lots of structural variants, they must be fairly small. We don't have any evidence of inversions. But again, we don't have a lot of power, so I couldn't exclude it.
Sure. Thanks.
No worries.
And then Sandy, you're next.
Okay, so Iris has also asked, well, we both asked kind of the same question, the second question, so I'm going to just combine both of mine into one.
Yep.
Is the fact that there were kind of these 3 introductions from the mainland, and the most recent one is from Ecuador and is the one that's invasive. Could that invasiveness be related to the fact that those populations in Ecuador are actually introgressed with Lycopersicum?
Well, that's a really interesting question. So let me just clarify one piece of our data, and that's that actually the invasive ones are somewhat less closely related to mainland populations than the 2 singular ones. So actually we think that introduction is older. So the invasive one. Yeah, so the one that the lineage that gave rise to all of those populations, that's
but still, if it comes from Ecuador, because when we were looking at all of the wild tomatoes, and Edith can probably say more about this than I can, is that we think a lot of those Ecuadorian populations are actually pimpinellifolium, which has been back-crossed with cultivated tomato. So that could be one of the reasons why it's more invasive.
That's a great question. And the answer is, I don't know. I haven't thought about it, but we could actually probably think about whether it has. So it's really complicated, as you know, because there's so much shared ancestral variation among these species that it's really hard to tease apart. But what one could do is say, is the invasive set of lineages, do they have more Lycopersicum ancestry than the other ones, right? And that may suggest something about
and you'd have to be careful about which Lycopersicum as well, because there's Lycopersicum, there's pimpinellifolium in many of the pedigrees of the cultivated tomatoes. So it's pretty confusing. But the other one, which Edith and I both asked, is that those original populations that Sarah identified, because her original observations of and I was hoping she was going to be able to join today, but I don't think she's been able to, but that's okay. I'm going to go see her next week or the week after. But her original observations of the potential introgression between cheesmaniae and pimpinellifolium came from the basura, came from the rubbish dump on Santa Cruz. And so are those 2 populations, MG114 and MG117, they're along that road that goes across Santa Cruz, and are they near the rubbish dump? Because of course the ones that were on the rubbish dump when Sarah looked at them would be gone because the rubbish dump has got much bigger.
Yes, one is them near the rubbish dump. I mean, yes. Yeah, I know. I mean, the work that she did was really amazing in terms of just its sort of comprehensiveness and in terms of the sort of detailed descriptions. And we couldn't have done what we did in terms of going back and thinking about this population.
Well, I didn't do it. Sarah did it. Sarah and Edith did it. I just kind of cheerleaded, really.
I mean, we really, that was enormously helpful to have that. But yes, one of those sites is from there. And it's one of the polymorphic sites. I don't remember.
And the other hybrid, the cheesmaniae galapagense hybrid, is also a very disturbed area. It's that kind of lagoon area.
It is.
The whole thing about the hybridization is happening where there's human disturbance as well, isn't it?
Without question, I think that that's true. I think that anthropogenic disturbance is definitely not helping the endemic species and may well be helping the spread of the invasives. It's not obvious to me why the anthropogenic effects would influence the phenotypic shift in fruit color, although there may be human preferences for that, I suppose.
Yeah,
but
I think that phenotypic thing is really cool, especially if it's happening near the basura, because it definitely wasn't happening in the basura when Sarah did her work.
Oh, awesome.
Mhm.
Good to know. Huh.
Leonie,
Hi.
I'm Iris. Nice to meet you.
Hi.
And well, regarding the commentaries by Sandy, I was in the Galápagos in 2008, and I saw this pimpinellifolium population along roads mainly. And I wonder if you have find this kind of putative hybrids inside the natural areas. I thought that they are mainly confining to disturbed areas. And another thing is that not only the fruits is a trait that can define this kind of hybrid. To me, this hybrids look like more than backcross to cheesmaniae, many backcross, because essentially the hybrids look more than like pimpinellifolium. And I wonder if here is playing a role the reproductive traits, mainly because cheesmaniae is mainly autogamous and very difficult the transfer of pollen between pimpinellifolium as a father to cheesmaniae as a mother. I don't know if I explain well, but I think to me the pollen flow is from the native tomatoes to the invasive plant. And also the plants are more vigorous in this kind of putative hybrids than the habit of the native tomatoes. I don't know if you saw that, or
Yeah,
this could
so
be related
yeah,
with
so
your
that's
finding.
yeah, a really interesting set of observations. Thank you for sharing that. So in terms of individuals that looked unusual from pimpinellifolium, those orange fruited individuals, we have some morphometrics of some of the leaf traits, and they don't look really like island species. They look like pimpinellifolium in those traits. It's really the fruit color that looks like the island species. For the ones that Matt collected, it may well be that we didn't see all possible hybrid. I mean, the field collections that Matt did were limited in terms of time and scale, and as you know, it can be difficult to get permissions to go to particular parts of islands and collect from particular parts of islands. So I'm sure that we missed cases where there were additional hybrids, and perhaps the cases that you're discussing look more like that. You asked about whether they're off the major roads, and the answer is we don't know. Wouldn't it be great to go down there for multiple months and tramp around and see whether there are other locations where pimpinellifolium is further away from human settlement? And the answer is we don't know. There's a few more remote sites that Matt did go to in 2019, but they're hard to get to, and it just takes time. So we were meant to be down there in June looking at more sites, and of course we couldn't be there because of the dreaded pandemic, but we hope to actually be able to go back down there and be more comprehensive about
Sarah and I once had this great idea. Sarah and I once had this grand idea of doing it, because the road goes north to south through Santa Cruz, and we had this grand idea of going orthogonal to that and going east to west and just basically walking the whole way across Santa Cruz, which would be really quite hard, but quite fun.
I mean, it's exactly how you would design a field survey looking for a rare and endangered species, right? You basically break it up into grids and go, okay, just go to the grid, go to the grid, go to the grid and see if it's there, rather than sort of going where it's been before or going where it's easy to get to. And that would be ideal. But as you say, logistically, it's pretty challenging, and it would require a fair bit of both monetary and other kinds of, you know, personnel support to be able to do it so comprehensively. Yeah.
We have a lot more questions. We'll see how far we can get through all this. But another big question from a couple of people is about the species Galapagense. Somebody asked, why is there no hybridization? Is it due to self-incompatibility? And then James is also talking about the same species, and then how could it be endanger the genetic integrity of the endemism?
Okay, so I think I can handle some of them. None of the red-fruited species are self-incompatible. They're all self-compatible. So they have all, yeah, they're all self-compatible. In terms of species barriers, in our hands, and in fact in other people's hands, there's not a lot of evidence of, you know, pollen-pistil interactions that are leading to rejection, or very strong pre-zygotic isolation, either among different accessions of the Galápagos species, or even with pimpinellifolium. There may be more mild post-zygotic isolation, but in terms of reproductive isolation, it's not really strong. So I suspect that there isn't more evidence of hybridization in the past just because there isn't contact. All right. So as Sandy pointed out, a lot of these places where these things seem to have at least initially started to hybridize are places that have where species have been brought into contact that weren't before. And so it's more about opportunity rather than sort of the presence and absence of species barriers. I think I answered the questions. I'm not sure.
I don't know if maybe Jamie can like add more, but he also asked about.
Hi Leonie, this is Jaime from Valencia.
Hi.
Brilliant talk. Yeah, I asked if when I was in Galápagos in 2000, I just saw red-fruited pimpinellifolium along the roads and disturbed sites,
right,
but not the orange ones. So this means that we are seeing evolution happening very quickly. And my question is, of course, the populations of Galapagense especially are very small, and with the large populations, at least I saw that with hundreds of individuals of pimpinellifolium, they produce pollen. Of course, they are mostly autogamous, but they can also, the pollinators bring pollen around and so on. So I was wondering if the genetic integrity of the small populations of Galapagense could be in danger because of recurrent pollination from pollen from pimpinellifolium.
Yeah,
So,
that's a great question. So part of the reason we first started working on that is that's a question that Maria had been interested in terms of hybridization between invasive guayaba and the native relative that it has on the islands, because there's that danger, right, of, you know, genetic swamping or whatever. So yes, I mean, demographic density has a huge effect on what kinds of directions of gene flow you might expect. Interestingly enough, we do infer with the HMM some evidence of gene flow from pimpinellifolium into cheesmaniae, but it's not as much. It's much more modest, but we have sort of fewer samples, so it's really hard to say. And it's actually, I mean, there's all sorts of reasons why you might not see it. One is that anybody who has gene flow from pimpinellifolium into you, perhaps that's not the greatest thing, and so maybe those individuals are not surviving as well. But it could be that they are, despite the fact that there's so much pollen around them from all of these large populations, they're still mostly selfing and therefore not at risk of getting integration from the invasive species. It's actually something we'd really love to understand better, because that's one of the reasons we started thinking about it. It's like, what's happening with these endemic populations, both in terms of just the persistence of the populations themselves, but their exposure to these invasives as well. So it's something we hope to be able to address further. But right now with our data, it's not obvious that there's a ton of gene flow in the other direction. A little bit, but actually not as much as seems to be happening into pimpinellifolium.
thank you. Excellent work.
No worries.
Congratulations.
No, thanks.
Maybe Stacy, do you want to ask your question? She might not be free so I can, okay, perfect.
Yeah, yeah, it was long-winded. I'm so sorry about that. But you won't be surprised it was about the color. And so, you know, I was thinking about the fact that, like, well, it's amazing that you were able to get down to some allelic variation that seems to be what's responsible for those orange fruits. And I think maybe that's possible because you know, what type of carotenoids you make is often related to those structural genes in the pathway, their expression or their sequence. But if it's a matter of whether you're making chromoplasts and thus carotenoids, you know, all those colored carotenoids at all, I think that might be more likely to be in that tangle of other genes that talk to the entirety of the carotenoid pathway. So in that context, I was wondering, what is known about the red-orange versus the green fruited? Like, does anybody match that? Is it gnarly, or is it like, oh yeah, we totally know which genes make you green versus red, yellow, orange?
The answer is, I'm sure somebody knows. I'm sure somebody knows. I don't. In fact, I mean, sure somebody else on this Zoom knows. I'm almost certain that the redness of pimpinellifolium, the genes underlying that, are pretty well understood in terms of their differences from the green fruited, but I don't know what they are. If Roger's on here, he probably knows. I don't know. But surely that is something. And actually, I don't know in terms of structural. So my guess is your question is whether it's structural. I don't know the answer to that question. Ah, there's Roger. Maybe Roger knows. You're muted, Roger.
Sorry about that. I'm not an expert on the carotenoids, but it's been shown that all the green fruited species have functional alleles at the beta locus, the beta lycopene cyclase. So that's one of the major ones. And they also have mutations at the R locus, the phytoene synthase. So at least those two are two major genes that have been mutated in pimpinellifolium.
Yeah. Yeah, so it's cool. I don't think about fruit color basically at all. Well, I hadn't until I started thinking about this particular case of fruit color. So I should probably know more about it.
I just think carotenoids are fascinating, and the more people learn about them, the more it seems just like super, super complex, you know, in terms of just the entire chromoplast differentiation. But which particular ones you make does seem to be pretty tractable. So it's like, here's another case of that, possibly.
Well, it's something I might have to think about. I might ask you about it.
Love to brainstorm, love to talk color.
Yeah, great.
So we have two more questions. The next one is Mattis, if he still on, wants to ask his question.
Yeah, sure. Sorry, I was confused. Hi, Leonie. It was absolutely a great talk. Thanks a lot.
Hi.
So I have a question about, the fact that, so you said that there's not so much, structural variation happening in wild species. So I was wondering if, what do you think, what's the way to get, variation in gene dosage and, potentially, changes, subtle changes in phenotypes that could help at having this equation in terms of phenotypes or other convergence. So I'm thinking if, do you think that heterozygosity, by example, can help at maintaining this variation in gene dosage within the population and potentially help at creating these new phenotypes?
Well, it's an interesting question. I think that it's really hard to maintain heterozygosity just passively, obviously, because you have segregation, right? So I think that the ways that I can imagine heterozygosity being maintained in natural populations is either because they are essentially compensating for a deleterious mutation, so they're essentially as a masking mutation. And I suppose you could have heterozygosity being maintained when you have a dominant effect, but you don't want to have two alleles, so you're like you're dominant lethal. I haven't thought much about heterozygosity per se in these populations. It's definitely true that island populations are extremely homogeneous. Some of the really early Allozyme work showed very little variation for within population, very little polymorphism in the population. We do see more polymorphism in these populations, both the endemic ones and the introduced ones, than sort of one might see with allozymes. But a lot of it is homozygous. There isn't tons of heterozygosity. So I don't know the answer to your question. It's interesting to imagine how one would even address it. But it's an interesting thought. How do you think?
I mean, I think maybe the part of the answer might be if you really see, I mean, hotspots or loci that remain, that are strongly enriched in heterozygosity, right? And I mean, clearly, I think it was the second B locus where you see, I mean, some of your lines quite a lot of heterozygosity, right? So the question is, there's just a transition, I mean, a transitional state to one side or the other, or is there a way, because maybe with interaction with the genes, I mean, epistasis, these kind of things, giving actually an advantage and natural selection towards the heads compared to the either the wild type or the mutated version, let's say.
Yeah, I mean,
but that's interesting.
Yeah, I guess it's testable by essentially comparing heterozygotes and homozygotes and thinking in terms of fitness. Testable in principle, testable in practice is hard. Yeah, it's an interesting thought. I guess my guess is that they're on their way. They're transitional. If you have a heterozygote, it's just because you haven't fixed it yet. But that's because I'm an optimist rather than.
Yeah, maybe I'm on the other side of this.
No, thank you.
But it's a good question. And okay, thanks for the answer. It was a great talk again, so really appreciate that.
Thank you.
And maybe one more last question, and then if people still have questions, they can email Leonie or ask her on Twitter if you want a whole group of people trying to help out and continue the conversation. So the next one is James. So James is on here still. Okay, cool.
Hey, thanks for the talk. I was just wondering, habitat-wise, do you think there's a difference in the background color between the habitats of pimpinellifolium in the native and the introduced habitat? Because in the photo you showed, the red fruits really sort of blended into the background to me, and the orange ones stood out a bit better. I was wondering if it was a contrast thing rather than maybe a color preference thing.
That's a great question, and that it gets at this idea that we don't. My global inference is that there's something good about being orange, but exactly what it is not clear. So changing your apparency would be one thing, right? That's essentially what you're talking about. It's not that they prefer, but it's just more apparent if you're orange rather than red. Yeah. The answer is there's so much habitat variation in mainland Ecuador. It's a little bit hard to imagine that there isn't some kind of background habitat that looks a bit like the Galápagos. I mean, there's lots of very dry lowlands, but it's not impossible. And so it could be that, or it could be that in an interaction between that and the pollinators that happen, the dispersers that happen to be present. So it's a really great question, but I think it still gets at, and it gets at this idea of like, what is it that could be good about being orange on the islands? And that is something that we're really keen to ask. So actually, I like that suggestion because it suggests that not only would you do a disperser preference experiment, but you might want to do it on a background that is more relevant to what a disperser might see on the Galápagos, rather than put it in a white bowl and say, oh, do you care? Because if they don't care, maybe it's not because they don't, it's because you're equally apparent when you don't put it on an appropriate background. So that's a great suggestion. I like that.
It would also be cool to map the fruits in the backgrounds in bird color space. Yeah, I guess. I mean, my understanding is bird vision is quite variable and depends on which species you're talking about. So that might be a bit. Yeah, unfortunately, there aren't that many species for which it's really been, for which you really have it. But it's, on the other hand, it's also, it depends on how closely related the dispersers are to, I guess, the models. But it's not that difficult to do, on the other hand. So. Well, I'm told in the chat, which is the first time I've caught up with it, that mockingbirds are almost certainly the main dispersers. Thanks, Sandy. And it may well be that there's a mockingbird that has a good model close to it. So that might be something we could think about. It's a great suggestion. Okay, cool. Thank you.
Thanks.
Thank you for your time, Leonie, and sharing your research. Next week is going to, there's a talk by Muhammad Ali. So he's going to be talking about medical compounds within Withania. So we'll send out an email. Thanks, everyone, though.
Thanks, everyone, for coming.
Thanks, Leonie. I sent you an email.
Oh, awesome. Great. I love information. Thank you. Thank you for the organizers. Bye.